Welcome back to my channel Only Bio, presenting up the last segment of the NEET 2020 paper from 81 to 90, all the MCQs with detailed explanation. Now, this question relates with the chapter Biotechnology. We have to match the columns, wherein column 1 has A as Bt cotton. I hope you will recollect that this Bt cotton has been quite frequently asked now. B is adenosine deaminase deficiency, C is RNAi, and D is PCR. Column 2 has first gene therapy, second cellular defense, third detection of HIV infection, and fourth Bacillus thuringiensis. And the options given are, the correct answer is 4. That is, Bt cotton can be definitely, you can guess this, it can be related with Bacillus thuringiensis. B, that is adenosine deaminase deficiency, can be related with the gene therapy. RNAi can be related with the cellular defense, and PCR can be related with detection of HIV infection. Now friends, we all know Bt cotton is genetically modified organism, which is a modified pest-resistant plant cotton variety that produces an insecticide to combat the bollworm. So, strains of bacterium Bacillus thuringiensis I had already mentioned earlier in some other question, wherein it produces 200 different Bt toxins and each one is harmful to different insects. So most notably, Bt toxins are insecticidal to the larvae of the moths, the butterflies, the cotton bollworms, etc. So the gene coding for Bt toxin has been inserted into cotton as a transgene, and this causes it to produce a natural insecticide in its tissues. Now the option B, that is adenosine deaminase deficiency, which is in simpler way called as ADA deficiency. This is a metabolic disorder that causes immunodeficiency. It is caused by the mutations in the ADA gene, and it accounts for about 10 to 15% of all the cases of autosomal recessive forms of severe combined immunodeficiency, that is called as SCID. Now, the enzyme adenosine deaminase is encoded by the ADA gene on chromosome number 20. And ADA deficiency is inherited in an autosomal recessive manner. This means the defective gene responsible for the disorder is located on an autosome, that is chromosome number 20. And two copies of the defective gene, one inherited from each parent, are required in order to be born with this disorder. The parents of an individual with an autosomal recessive disorder both carry one copy of the defective gene but usually do not experience any signs or symptoms of the disorder. That is, they act as carriers. Okay, so you'll have to remember this deficiency can result into buildup of DATP in the cells, and developing T cells and B cells, which are the most mitotically active cells, are highly susceptible to this particular condition because this prevents the DNA synthesis process. Okay, then the third option, that is cRNAi. Here इसका reference अपने books में नहीं है, so please ध्यान से सुनिएगा. RNAi, that is RNA interference. I stands for interference. It's a biological process in which RNA molecules inhibit the gene expression or translation by neutralizing the targeted mRNA molecules. Historically, RNAi was known by other names, including co-suppression, या post-transcriptional gene silencing and quelling. Now, because the discovery of RNAi and its regulatory potentials, it has become evident that RNAi has immense potential in suppression of desired genes. Okay, so this is what is RNAi. It is a valuable research tool both in cell culture and in living organisms. The option D, that is PCR, it's a method widely used to rapidly make millions to billions of copies of a specific DNA sample, which allows the scientist to take a very small sample of DNA and amplify it large enough in the great quantity to study in details. You might be remembering this technique was invented by American biochemist Carrie Mullis. Now, PCR is used to detect HIV's genetic material, that is RNA. And these tests can be used to screen the donated blood supply and to detect very early infections even before when the antibodies have been developed. This test can be performed just days or weeks after exposure to HIV. Now, the next question relates with the chapter Kingdom Planting. The question is, Floridean starch has structures similar to dash. A. Amylopectin and glycogen. B. Mannitol and algin. C. Laminarin and cellulose. And D. Starch and cellulose. And the correct answer for this question is A. Amylopectin and glycogen. Friends, remember Floridian starch is a type of storage glucan found in glaucophytes and in red algae, which are also known as rhodophytes. It is found in grains or granules in the cell cytoplasm and is composed of an alpha-linked glucose polymer with a degree of branching intermediate between amylopectin and glycogen. So this is the right answer here. Okay. Then coming to the option B, mannitol. That's a type of sugar alcohol. Okay, which is mainly used as a sweetener, you might have heard of. It is also used as a diuretic for producing, forcing urine production wherein people are suffering from kidney failure. Algin is a type of carbohydrate that is found in brown seaweeds and it is also produced by some bacteria. And algin is notably used to lower the cholesterol levels and to reduce the amount of heavy chemicals including strontium, barium, tin, cadmium which are taken up by the body. Okay, so algin is also used up for the prevention and treatment of high blood pressure. That's the other use of it. Now the option C wherein we have laminarin. Okay, this is also storage glucan, a polysaccharide of glucose which is found in brown algae and it is used as a carbohydrate food reserve in the same way like chrysolaminarin is used by phytoplanktons. Then cellulose, laminarin के साथ जो option है cellulose. We all know it's a polysaccharide that consists of linear chain of several hundred to many thousands of D-glucose units, and this cellulose is a primary component of the cell wall of green plants and in few forms of algae and the Oomycetes. Now starch, or starch which is also called as amylum. This is also a carbohydrate which consists of glucose units joined by glycosidic bonds. So in this we come to the answer A. Now the next question relates with the chapter hormonal as well as genetic engineering, and the question is, which of the following statements is not correct? Not correct, that is wrong. A. The proinsulin has an extra peptide called a C-peptide. B. The functional insulin has A and B chains linked together by hydrogen bonds. C. Genetically engineered insulin is produced in E. coli. And D. In man, insulin is synthesized as a proinsulin. And the correct answer is B. Friends, please remember insulin consists of two polypeptide chains, the A and the B chains, that are linked by disulfide bonds. It is, however, first synthesized as a single polypeptide chain called as the pre-proinsulin in the beta cells and which forms proinsulin. In the rough endoplasmic reticulum, the proinsulin folds into the correct conformation and three disulfide bonds are formed. So about almost 5 to 10 minutes after its assembly in the endoplasmic reticulum, proinsulin is transported to the Golgi network where immature granules are formed. Proinsulin undergoes maturation into active insulin through the action of cellular endopeptidases known as prohormone convertases and some other enzymes. So this releases a fragment called as the C-peptide, and this leaves, once the C-peptide is released, we are left out with two chains A and B which are linked by disulfide bonds. So the C-peptide chain is the central portion of proinsulin and the primary sequence of proinsulin goes in the order B, C, A. matlab the B and the A chains are connected via C. Please remember, इनमें से B and A chains were identified on the basis of the mass and C peptide chain was discovered later. The next question relates with the chapter evolution, and the question is, the flippers of penguins and dolphins are examples of A. Convergent evolution, B. Industrial melanism, C. Natural selection, and D. Adaptive radiation. And the correct answer for this question is A. Convergent evolution. Convergent evolution is when different organisms, when two species with different ancestral origins develop similar characteristics. Opposite to this is divergent evolution, wherein two species diverge from a common ancestor and develop different characteristics. Okay, another example that we can give, that is a textual example for convergent evolution, is the similar nature of the flight or the wings of insects, birds, and bats, etc. Right? All these have the same function and are similar in structure, but each evolved independently. Okay, now the option B, that is industrial melanism, that is the darkness of the skin or the feathers or the fur acquired by a population of animals living in an industrial region where the environment is darkened by the soot. Okay, many animals have a body color that allows them to blend in with their background. Okay, and this is something which is also related with industrial melanism. Now the option C, that is natural selection, this is the process through which populations of living organisms adapt and change. Individuals in a population are naturally variable, they change, right? And they change in different ways. Okay, that is for adjusting to the climate. And the fourth one, that is adaptive radiation. It is a process in which organisms diversify rapidly from an ancestral species into a multitude of new forms, particularly when a change in the environment makes new resources available, creates new challenges, or opens new environmental niches. For example, the alterations in the beaks of finches of Galapagos Island. The next question relates with the chapter Evolution of Life again, and the question is, which of the following refer to correct example or examples of organisms which have evolved due to changes in environment brought about by anthropogenic action? A. Darwin's finches of Galapagos Islands. B. Herbicide resistant weeds. C. Drug resistant eukaryotes. D. Man created breeds of domesticated animals like dogs. And the options are A and C, B, C and D, C only D, and D only A. The correct answer is B, that is B, C, and D. Now, friends, what exactly do we mean by anthropogenic activities? Anthropogenic activities are those human activities which have resulted into increased pollution of soil all over the world. And these pollutants can be either organic or inorganic, including various heavy metals. Okay, now इसमें जो option जो दिया हुआ है, if we relate this, we can easily understand the answer is B, C, and D. These are the, what you can say, repercussions of anthropogenic activities of human being, right? The next question relates with the chapter Human Respiration, and the question is, identify the wrong statement with reference to transport of oxygen. A. Partial pressure of carbon dioxide can interfere with oxygen binding with hemoglobin. B. Higher H plus concentration in alveoli favors the formation of oxyhemoglobin. C. Low partial pressure of carbon dioxide in alveoli favors the formation of oxyhemoglobin. And D. Binding of oxygen with hemoglobin is mainly related to partial pressure of oxygen. And the correct answer is B. Friends, partial pressure is the pressure of a single type of gas in a mixture of gases. And this is extremely important in predicting the movement of the gases. See, gases tend to equalize their pressure in two regions that are connected. So, a gas moves from an area where its partial pressure is higher to an area where its partial pressure is lower. In addition, the greater the partial pressure difference between the two areas, the more rapid is the movement of gases. Now, all this in connection with the respiration process. Let's understand the option A. The partial pressure of carbon dioxide and oxygen, as well as the oxygen saturation of hemoglobin, it influences how readily hemoglobin binds with carbon dioxide. The less saturated hemoglobin is and the lower the partial pressure of oxygen in the blood is, the more readily hemoglobin binds to carbon dioxide. So this becomes a correct statement. And you can relate this up with the Bohr's effect. You can study this in the chapter respiration, right? And the others are from this explanation. You can already relate that Bohr's effect, it refers to the shift in the oxygen dissociation curve, and that's caused by the changes in concentration of carbon dioxide or the pH of the environment. So you can relate this up and we come to the correct answer. That's B. That is the wrong statement. But the next question relates with the chapter plant growth, and the question is, the process of growth is maximum during dash. A. Lag phase. B. Senescence. C. Dormancy. D. Log phase. And the correct answer is log phase. Here the question refers to the seed germination. Early seed germination begins with imbibition of water and can be described by increase in the seed fresh weight due to uptake of water. And these phases are characterized by rapid increase in water during the imbibition phase, followed by a period where there is little water uptake, and this is what is the typical feature of lag phase. Now the second option, that is plant senescence, is the process of aging in plants. These are both stress induced and age related developmental aging. The chlorophyll degradation during leaf senescence reveals that the carotenoids such as anthocyanin and xanthophylls is the cause of autumn leaf color in deciduous trees. The option C, that is dormancy, it is a period in an organism's life cycle when growth or development, even in case of animals, the physical activities are temporarily stopped. And this minimizes metabolic activity and therefore helps an organism to conserve energy. Dormancy tends to be closely associated with environmental conditions. And the fourth option, that is the log phase, which is called as the logarithmic phase or the exponential phase, is the period that is characterized by cell doubling. This question relates with the chapter organisms and environment. The question is, which of the following regions of the globe exhibit highest species diversity? A. Madagascar, B. Himalayas, C. Amazon forests, and D. Western Ghats of India. And the correct answer for this question is C. Amazon forests. Friends, species diversity is greatest in the tropics, particularly in the tropical forests and coral reefs. The Amazon basin in South America has the largest area of tropical forests. Now, there are reasons why there is species richness in this particular area. First, it receives more solar energy. So, more the solar energy, it results into high productivity and high species diversity. Also, it has less seasonal variations. So, it has more or less constant environment, and this promotes the niche specialization and thus high species richness. Now, why is Amazon forest so important? Other reason is this Amazon rainforest plays an important part in regulating the world's oxygen and carbon cycles. It produces roughly 6% of the world's oxygen and has long been thought to act as a carbon sink, meaning it readily absorbs large amounts of carbon dioxide from the atmosphere. The next question relates with the chapter biotechnology. The question is, the sequence that controls the copy number of the linked DNA in the vector is termed as dash. A. Oricide, that is the site of origin. B. Palindromic sequence. C. Recognition site. And D. Selectable marker. And the correct answer for this question is A. Oricide. Now first let's understand what does copy number mean. The copy number refers to the average or expected number of copies that can be produced per host cell. Now plasmids are either low, medium or high copy number. Now, plasmids also vary widely in the copy number depending on some factors like the site of origin and its constituents, the size of the plasmid and its associated insert, and the third is the culture conditions. Now, origin of replication is a sequence from where the replication starts, and any piece of foreign DNA is linked to the sequence. The replication occurs inside the host cells, and this new sequence is also responsible for controlling the copy number of the linked DNA. So therefore, if any person wants to produce many copies of the target DNA, then one should clone in a vector whose origin gives support to high copy number. Now, palindromic sequence you might be aware of. This is a nucleotide sequence in a double-stranded DNA or RNA molecule wherein reading in a certain direction, that is Y dash to 3 dash on one strand, matches the sequence reading in the same direction on the complementary strand. Okay, and you might be knowing recognition site is nothing but the site which is recognized by the nucleotide sequence composed typically of 4, 6 or 8 nucleotides by the restriction enzymes. And selectable marker is a gene that can be introduced into a cell, especially in a bacterium or to the cells in the culture, that confers a trait which can be used for artificial selection. The next question is taken from the chapter Human Reproduction, and the question is match the following. We have two columns, column 1, column 2. In column 1, A is placenta, B is zona pellucida, C is bulbourethral glands, and the D is Leydig cells. Okay, and column 2, we have androgens, human chorionic gonadotropin, third one is the layer of the ovum, and fourth one is the lubrication of the penis. And these are the options given, and the correct answer is 3. That is, A, placenta is related with human chorionic gonadotropin hormone, B, zona pellucida is the layer of the ovum, C, bulbourethral glands help in lubrication of the penis, and Leydig cells are related with androgens. Now, placenta क्या होता है? Placenta is an organ that develops in the uterus during pregnancy, and this provides oxygen and nutrients to the growing baby and also helps in removal of the waste products from the baby's blood. Okay, the placenta attaches to the wall of the uterus and the baby's umbilical cord arises from it. Now, HCG, a human chorionic gonadotropin, is a hormone produced by the cells that surround the embryo, that is syncytiotrophoblasts initially, which eventually forms the placenta after implantation. So, presence of HCG is also used up as a test to detect up the pregnancy. Now, this hormone stimulates the corpus luteum to produce progesterone to maintain pregnancy. Now, the option B, zona pellucida. This is a specialized extracellular matrix surrounding the developing oocyte, that is the ova, within each follicle within the ovary. And this is thought to be formed by secretions from the oocyte and the follicular granulosal cells. And in human oocytes, it consists of 4 types of glycoproteins, namely ZP1, ZP2, ZP3, and ZP4, and each has a role in fertilization. And it is zona pellucida has many roles. It helps in oocyte development, protection during growth, transport, fertilization, binding with the spermatozoa, preventing polyspermy, helping in the development of the blastocyst, and preventing premature implantation. Now, the option C, that is bulbourethral glands, which you might be aware, are also called as the Cowper's glands, right? These are a pair of P-shaped exocrine glands located on the posterolateral side of the urethra, and this contributes to the final volume of the semen by producing a lubricating mucus secretion. In response to sexual stimulation, it produces an alkaline mucus fluid. And the option D, that is Leydig cells, which are also known as the interstitial cells of Leydig. These are found adjacent to the seminiferous tubules in the testicles, and these are important because they produce testosterone in presence of LH. Friends, like, subscribe, and hit the bell icon if you haven't. Thank you so much.